that we employed for the glycoproteomics through high resolution LC-MS or mass spectrometry connected to an LC system for the characterization of glycosylation on SARS-CoV-2 spike protein. So this procedure can be employed for any proteins or glycosylated proteins, and in glycosylated protein mostly the differences happens in the case of data analysis. In order to analyze the glycosylation on the spike protein, the proteins from the viruses were digested by enzymes or proteases such as trypsin after protein denaturation and alkylation. Protein denaturation is uncoiling of proteins from their tertiary or quaternary structure, and alkylation is the blocking of the cysteine. So in denaturation, the cysteine disulfide bonds will be cleaved, and these cysteine disulfides can form back if we don't block the SH or sulfhydryl group of cysteine. So we react with an alkylating agent, and this alkylation procedure will block the reformation of disulfide bridges. Then the proteins will be in an uncoiled form. So these uncoiled protein will be digested by enzymes, and then we will get a mixture of peptides and glycopeptides. So these mixture of peptides and glycoproteins are later injected to an LC-MS instrument or mass spectrometry, and mostly these days most labs employs high resolution mass spectrometer for this type of analysis. And then later these LC-MS data files, once you obtain that, will be searched against databases of both proteins as well as glycans using specialized softwares such as Bionic, which is one of the leading software in glycoproteomics right now, and the glycosylation on the protein is determined. So later these spectra will be manually evaluated for the peptide fragments and glycan fragments. I have an example later I will show in another slide. And then the glycan structures on the proteins are interpreted. So this is the N and O glycosylation found on the original strain, that means the wild type strain of SARS-CoV-2 spike protein through the glycoproteomic strategy. So we were the first group to report this glycosylation on SARS-CoV-2 in early 2020, particularly the O glycosylation, which was not expected in the coronaviruses. Further, the relative ratios of glycans at each sites were determined and quantified. So you could see all the 22 sites on SARS-CoV-2 spike protein, and each site are modified by different types of glycans and different numbers of glycans, and they are carrying in a different relative ratio. So this information will provide an overall information about how these glycosylations are more present in a given spike protein. So different types of glycosylations such as high mannose and complex type N glycans are detected on each of the SARS-CoV-2 spike protein as shown in the figure. The N glycosylation and O glycosylation at the receptor binding domain, which actually binds with the human receptors such as human angiotensin converting enzyme, are most important. This is because the alteration in the glycosylation at these sites can affect the viral infectivity. This figure shows the two subunits S1 and S2 of SARS-CoV-2 spike protein and different types of N glycans color coded based on the type of N glycans, and the region where O glycosylations are present or reported on spike protein. So this is a representative MS fragmentation mass spectrum of a glycopeptide. In this case, this is an O glycosylated glycopeptide, and this spectrum provides rich information about the peptide sequence, glycans attached to the peptides, and also the sites of glycosylation. We also detected specific glycan ions and fragments and confirmed the presence of glycans. You could see here these pink color dots or diamonds shapes are sialic acids. So those are showing some typical masses of 274. So those indicates the presence of sialic acid in this glycopeptide MS spectrum. The yellow color squares are galactose, N-acetyl galactosamine. So those are showed in yellow color squares, and those shows the presence of such type of glycans in this spectrum. So similarly, now we confirm the presence of glycan fragments, and same way the peptide fragments such as B and Y ions of the peptides are also found in this particular spectrum. Then there are several fragments which comprises both peptide and glycans. You can see that peptide with a yellow squares or then elaborated glycans. So that is another set of information that we can get from this spectrum. And the next part is neutral losses. When the fragmentation happens in the mass spectrum, the glycan chains are broken in a sequential manner. So first the terminal sugars, those pink diamonds are cleaved in a sequential manner, and we will see the mass differences. So those are called neutral losses. So glycan neutral losses will also be observed in this spectrum. And the final information is about the site of glycosylation. So for example, in this peptide of VQPTES, so there are two possible locations of O glycosylation. We could conclude in this case that the glycosylation happened on the position T by obtaining this fragment Y7 plus glycan. So we did not observe a fragment of Y5 or Y4 with a glycan. That means Yn starts from this R328. We are not seeing any fragment of Y5 or Y4 with a glycan. We are seeing a fragment of Y7 with a glycan. So that indicates the possible presence of the glycosylation on this T.